Hello everybody. Today we are going to begin our study of DNA, which is the molecule of heredity. DNA stands for deoxyribonucleic acid. DNA is located in the nucleus, not in the cytoplasm, of eukaryotic cells. So it is the material inside this structure. And in eukaryotes, it forms a complex with proteins called chromatin. You may remember that in prokaryotes, there is just circular DNA. It's a far simpler system. And what we want to look at today is, how do we know that DNA is the hereditary molecule? Well, a series of elegant experiments done early in the last century established that fact. There was the Griffith Experiment that was published in 1928, the Avery et al. Experiment in 1944, and the Hershey Chase experiment in 1952. And we're going to look at those experiments today to see how we learned that DNA carries the information of heredity. So let's start with the Griffith experiment, since it was the earliest. What Griffith knew was that there were two strains of the same bacterium, and he called them the R and the S strain. The R strain, which stood for rough because it looked rough in the microscope, is benign, meaning that it doesn't cause a disease. Whereas the S strain, which stood for smooth, and you could see that in the microscope, is virulent, meaning it causes pneumonia. And Griffith used mice as his test animals. It was also known that you could kill the bacteria with heat. So if you had either strain and you heated it to a certain temperature, the bacteria would die. So Griffith did the following experiment. He had 4 treatments. First, he injected mice with the R strain, the rough, benign strain. Second, he injected mice with the S strain, the smooth, virulent strain. Third, he injected mice with heat-killed S strain. And fourth, he injected mice with heat-killed S strain plus the R strain. So what were the predicted results of this experiment? Well, for the injected mice with R strain, he thought that nothing would happen. For the injected mice with S strain, he thought that they would die because they would get pneumonia and die. For the injected mice with the heat-killed S strain, he predicted that they would survive because the bacterium was no longer alive and so couldn't be virulent. And finally, he thought that mice injected with the heat-killed S strain and R strain simultaneously would survive because heat-killed S strain and R strain alone were benign. Let's see what happened. Here are the 4 tubes. The rough strain, as predicted, the mouse lived. The smooth strain, as predicted, the mouse died. The heat-killed smooth strain, as predicted, the mouse lives. And with the rough and heat-killed strain given simultaneously, wait, there's an incongruous result. The mouse dies. So I recommend that you make a table of this experiment and write down the results in tabular form so you understand what happened. Griffith's conclusion is that some substance in the heat-killed virulent strain transformed the non-virulent bacterium into the virulent form of the bacterium. So what was this substance? People had a hunch that whatever transformed the bacterium was the same thing that provided hereditary information. And so there was great debate as to what molecules would be the hereditary molecules. And people mostly thought it must be proteins, DNA, or RNA. Guess which one most people thought was the hereditary molecule? If you said DNA, you're 100% wrong. If you said RNA, you're also wrong. Most people thought it was proteins. And why did most people think it's proteins? Because proteins have obvious functions in the body, and at that point, there was no known function for DNA. Perhaps that should have been a clue. And not only that, but proteins have 20 different component amino acids. So they have more letters, so to speak, than DNA, which only has 4 different types of nucleotides in it. So Griffith showed that there was some molecule that was able to transform bacteria, and that this molecule was probably the same as the hereditary molecule. Along come Avery et al. In 1944, and what they did was they took heat-killed virulent form and they removed all the carbohydrates and fats from it. So they knew how to grow a bunch of virulent form, kill it with heat, and then remove all the carbohydrates and fats. That means they were left with RNA, DNA, and proteins. And they divided that solution into 3 tubes. In one tube, they placed enzymes that would destroy proteins, but it would leave the DNA and the RNA. In another tube, they placed enzymes that would destroy RNA, but would leave the DNA and protein intact. And finally, in another tube, they placed enzymes that would destroy the DNA and leave the RNA and protein intact. So what are the predictions? The predictions are, if tube 1 does not cause transformation, then proteins are the hereditary substance. If tube 2 does not cause transformation, then RNA, which was destroyed in that tube, would be the transformative agent. And in tube 3, if that did not cause transformation, then it would be DNA. That was the hereditary and transformative agent. Let's look at what their results showed us. Here are the heat-killed S cells. They remove the lipids and the carbohydrates. They divide it into 3 tubes. They add the enzymes, proteinases, get rid of the proteins. Ribonuclease gets rid of the RNA. Deoxyribonuclease gets rid of the DNA. And then they use these solutions to see whether they can transform cells. And don't forget, all they have to do is grow the cells on a bacterial plate and look at them to see whether they become smooth or not. And in only one case did they not get transformation. In this case, where they removed proteins, they got transformation, which means that protein couldn't have been the transformative agent. In the second case, where there was no RNA, they got transformation, so that rules out RNA as a transformative agent. Thus, they think DNA is going to be the transformative agent, and indeed, in this case, they get no transformation occurring, no S cells appear. So now, Avery et al. Can conclusively say that DNA is the transformative agent. Finally, in 1952, Alfred Hershey and Martha Chase published the following experiment. They used viruses that are known to attack bacteria, and they only attack bacteria. They're called bacteriophages, which literally means eaters of bacteria. These viruses consist only of a protein coat and a DNA core, and that was known. Thus, they provide a great way of further confirming whether DNA or protein is the molecule of heredity. They act by taking over the host cell to make more virus. So they grew 2 populations of virus. One strain was grown in bacteria that were supplied with radioactive phosphorus. So don't forget, the way a virus works is it takes over the bacteria's machinery and makes it make more copies of itself. And these bacteria are grown with radioactive phosphorus. And the reason you use radioactive materials is because you can follow it. You can find it later on. As it moves from organism to organism, you can use like a Geiger counter to see where the radioactive phosphorus is. On the other strain, they grew the virus on bacteria that were supplied with radioactive sulfur. So why did they do that? Well, it's because phosphorus occurs in DNA and not in protein. So the strain grown on radioactive phosphorus had radioactive DNA. And sulfur occurs only in protein and not in DNA. So viruses grown with radioactive sulfur will have radioactive proteins. They added each strain of virus to separate colonies of bacteria. And then they used a series of tools that involve centrifugation, and I won't go into the details here, to look to see what went into the cells and what didn't go into the cells. Was it the protein that went into the cells, or was it the DNA? Their prediction was that if it was the protein, then they would find radioactive sulfur inside the cell. And if it was the DNA, they would find radioactive phosphorus inside the cell. So what did they find? And how did they do this amazing experiment? Well, here it is. They grew the bacteria, then they put the bacteria in a blender, and they just gently agitated it, and that knocked any attached viruses off the cell. And then they put the solution in a centrifuge, spun it at very high speed, and a solid mass forms at the bottom called the pellet, which is the remains of the bacterial cells. And they did this with both cells that had been exposed to radioactive sulfur or radioactive phosphorus. And here's what they found. When sulfur was the radioactive substance, they found no radioactivity in the pellet, only in the liquid above, meaning the sulfur had never gone inside the bacteria. When phosphorus was a radioactive agent, they only found phosphorus in the pellet, and they didn't find it in the supernatant, the liquid on top. This suggests that phosphorus went inside. Once again, it suggested that DNA was what was causing the cells to make copies of the virus. It wasn't the protein that was reprogramming the cell, it was the DNA. So, Hershey and Chase's conclusion, DNA is absolutely the component of the bacteriophage that is injected into the cells, and the protein coat remains outside and can't be the agent of heredity that takes over the cellular machinery to make new viruses. So, that's it for today. I want you to review these experiments until you understand how they showed that DNA is the transformative and hereditary molecule. Next time, we will go on to look at the structure of DNA. Until then, go outside, look at nature, ask biological questions, and I'll see you soon to talk about them.